Once we work out how to sequence amino acids, what would we want to do next? So biochem is really concerned with proteins and enzymes. It's really, for us, we're really going to be concerned with two things. DNA, the stuff that codes for proteins and enzymes and structural and functional proteins. It's the DNA that codes for it and the enzymes that it produces. And when you want to study a protein in detail to figure out how it's catalyzing a reaction, because most drugs do one of two things. They either up-activate an enzyme or down-activate it. Let's say there's an enzyme that's killing you. You want to stop that. If there's an enzyme there's enzymes that could be killing you. For example, there's an enzyme, E3, that's a ubiquitinating enzyme that's there specifically to destroy other proteins, but sometimes it goes on a rampage and destroys things that it shouldn't destroy. So what if it destroys tumor suppressor proteins? So now you have nothing to stop tumors from popping up. So sometimes you want to up-activate an enzyme, sometimes you want to down-activate an enzyme. How do you do any of that? You need to study the enzyme completely. You need to know. So step one is to study its amino acid composition. Step two is to study what its two-dimensional structure is, which is alpha helices and beta pleated sheets. Step three is to study how it folds, and then how its functional groups go into an active site and how it catalyzes reactions. All of that is for biochem. I mean, I think we went far enough in this chapter that anything beyond it we could cover in an actual biochem course. So we're switching into the next chapter, which is nucleic acids. Trust me, I won't leave you hanging on the remainder of amino acid and how they combine to make proteins and all that, but that's for the full biochem course. I'm very happy that we covered Henderson-Hasselbalch and some buffer ideas. There's much more. There's much more. We only scratched the surface. Here's a general rule. You're starting to get in the right place with Henderson-Hasselbalch and buffers when you come up to me or your professor and start hysterically crying that you think you're never going to learn Henderson-Hasselbalch. You're finally understanding its complexity, and now you're close. If you haven't started crying about it yet, then you're very far away, is what I'm telling you. But once you start crying, don't artificially start crying. It won't help. But once you start crying because you realize, oh my God, there's some questions that's almost like, solve this question. And that's it. There's almost no question. And you're like, how the hell, there's no data. Like, what am I supposed to do with this? That's when you start crying. And then you realize there's some convoluted Henderson-Hasselbalch related questions. I'm glad we kind of started on that journey. So we're not going to be totally lost at level 1. We can draw titration curves. We can draw relative fraction plots. We're not zero on that. Get into nucleic acids. So what are nucleic acids? They're DNA and RNA. Yes, that's good. That's good. So a nucleic acid is a polymer. This is a polymer. What's the monomer? The monomer is the part of the polymer. What's a polymer? It's made out of monomers. What's a monomer? The part of the polymers. Goes in a circle. Many what? Many nucleotides. So a nucleotide is the individual unit. They link up in the chain to give you the polynucleotide or the nucleic acid, right? It's interesting that it's called a nucleic acid, or it's called deoxyribonucleic DNA, deoxyribonucleic acid, when really it's its own conjugate base under physiological pH. Like, it's a nucleic acid, but the phosphate groups are not protonated. They're negatively charged because they have a very low pKa, so at a pH of 7.2 to 7.4. So should we rename it to deoxyribonucleic conjugate base? What's a nucleotide? Well, a nucleotide is but what is it? It's a nucleoside attached to a ribose. That's not right. It's a nucleoside with a phosphate. That's more correct. That was a brain glitch I just had. It's been a long day. It's a Monday. It's bad weather outside. So what's a nucleoside? A nucleoside is a nitrogenous base plus a ribose. A nitrogenous base attached to the N-americ position of a ribose in an N-glycosidic bond, specifically an N-ribosidic bond. Any idea of what I just said? Who has an idea of what I just said? Half the room. Some of us are shaky. It's a Monday, it's bad weather. Nitrogenous base can be what? It could be adenine, guanine, cytosine. I need more space. Nucleoside plus phosphate makes a nucleotide. A nucleotide links up in a polymer called a nucleic acid, right? And a nucleoside is a nitrogenous base plus a ribose linked like this. Right? In an N-ribosidic bond to carbon 1, the N-americ carbon. Can we follow that? Could you take this apart? Can we remove that? Put an OH there, realize you're looking at the hemiacetal. Can you undo the Hayworth and figure out what the stereochem is? Ribose is like a 5-carbon allose. Because allose is a 6-carbon allose, because that's the name allose and why it's the name allose. Anyway, so what are the nitrogenous bases? You have, once again, adenine, guanine, cytosine. Every time I say that, I think sight unseen, but that's nothing to do with anything. Cytosine, uracil, and thymine. Now, that's the nitrogenous base by itself. It's called adenine. By the way, how many of you know there's more than this, nitrogenous bases? There's inosine, there's pseudouridine, there's ribothiamine, there's many more. It's just these are the common ones that you find in DNA or RNA, and then there's other ones that are in like tRNA and other places. So there'll be more than this. But when adenine, the nitrogenous base, is linked to a ribose, the name now reflects the fact that it's a nucleoside. So this whole thing is a nucleoside. So the base itself is called adenine, but when it's a nucleoside, it's not adenine, it's adenosine. Bless you. Someone sneezed. So adenine is adenosine, and guanine is guanosine. This is easy, you got this. Cytosine is, it's not cytosine, so what's cytosine? It's not cytosinosine. Cytidine. Uracil is not uracilocene or anything like that, it's uridine. Uracil. Sounds like urine. Does that have anything to do with it? That's the nitrogenous base uracil. What do you see over here, in case you know what that is? That is a urea, actually. If I have a carbonyl with 2 NH2s on the sides, that's a urea. So maybe it has something to do with the urea cycle. Maybe it just has a urea in it, so it's called a uracil. Uridine. What does thymine? Thymidine. Why are they trying to confuse us? They're not. They're not. The reason that these are called scenes and these are called dienes is because a couple of these are purines and a couple of these are pyrimidines. So these two are purines and these three are pyrimidines. Pyrimidines. In case you're confused and need some way to remember it, remember to cut pi. CUNT are pyrimidines. It gets a little better. In biochem, you always have the small name is the bigger thing, and the bigger name is the smaller thing. If you take a look at the structure of these, some of these nitrogenous bases have two rings connected to each other, and some of them have just one little tiny old ring. Pyrimidines is one little ring. Well, purines, small word. Two rings, right? It's how backwards. But now it's nice. Because what's a pi? When you cut a pi, is a pi, or is a pi some weird mutant pi? You see, that's a pi, right? This helps you remember to get. If you can't remember can't remember CUT as pyrimidines, you got bigger problems. You know, like. No, I'm saying like you've been through Orgo 1 and you've been through Orgo 2, and there are some schools that for Orgo 2 ask you this on a final exam: which one of these functional groups is called aldehyde? You know what I'm saying? Like there's the Talens test, there's so much stuff. And if somebody can't recognize the aldehyde functional group, they got bigger issues in terms of chemistry, is what I'm saying, then you should by default not need to remember this. But if you're on an exam and some nervousness happens, forget how to spell your name. That happened before, may have happened to me. I'm not crying about it. Maybe CUT cut pi can help. But that helps realize why some things are deans and some things are scenes. And do you see? Hopefully you see. So RNA has A, G, C, and U, and DNA has A, G, C, and T, and why? What's the difference? So RNA is a ribose base, and DNA is a 2' deoxy ribose. Gained another follower. These things are moving along now. Things are happening. Okay. What came first? The chicken or the egg, right? Like, what came first, DNA or RNA? Do we have an answer for the other question? Not this. What came first, the chicken or the egg? Let's take a vote. Who said it's the egg? 2 people. Who said it's the chicken? More people. So how could a chicken come first before an egg? No, but how could a chicken come before an egg? For it to be really, truly called a chicken, it should be born of a chicken egg. It could be the first chicken-looking thing, but it's not really a chicken until it's born of a chicken egg. How can an egg be a chicken egg if it's not coming from a chicken? There's actually, I think, a simple answer to this. Because it could be an egg, it's just not a chicken egg. So there's a simple answer to this. There's no actual thing that we call a chicken as long as we believe in evolution. Because then there is a gray morphing area. There's no cutoff where you say, that's the chicken. Do you see what I'm saying? Come back 100,000 years, the chicken we'll see is not going to look anything like today's chicken. And when people say, what came first, chicken or the egg? We'll be like, that's not even a chicken. You know, so evolution says that there is no Platonic form of a chicken. Have you ever heard of the Platonic form, Plato's forms? If you haven't, read what I'm talking about. back to this. What came first, RNA or DNA? Is it the same kind of nonsense story? No, actually, one of these came before, and it's misleading. It's confusing if you take a look at how transcription and translation happens. We have DNA, at least in eukaryotic cells, in the nucleus, and you don't want to like mess with the DNA, so you make a copy of it, and that's called transcription. Your DNA gets copied into RNA. There's 3 types of RNA: messenger RNA, ribosomal RNA, transfer RNA. Well, you call this transcription. It makes a transcript, copy. And then this goes over to the ribosome and it does translation. And it makes a protein, a peptide, an enzyme, so on and so forth. So just by looking at it this way, you would think that, well, DNA had to come first because DNA gets copied to RNA and then RNA gets translated. But actually, that's not at least what the evidence points at. It actually points at RNA coming before DNA. There's an RNA world hypothesis that simple organisms didn't need anything that was kind of deeper storage. But as life gets more complex, you kind of need more complex storage. So the first computers had some kind of floppy drive. They didn't even have a floppy. They had punch cards. Those are the first computers. Then they had floppy drives. Then they had hard drives. Then there's cloud storage. And like now, you might download something from the cloud. That doesn't mean the cloud came first. Before, the more complex you need, the better storage system you need. So we're going to look at the differences between RNA and DNA and see what evidence we could point at as why we say RNA comes before DNA. First, the ribose itself, right? So this would be the structure of ribose. I can't explain to you all of the things behind the RNA world hypothesis, because in order to do that, we're going to have to go and look at biological pathways. We're going to have to look at glycolysis and how it happens in nearly all organisms in the cytoplasm. And then say, why is that significant? And then take a look at the NAD and NADH that you have in glycolysis and the ADP and the ATP and see if they're ribose-based or deoxyribose-based, and start seeing what things were prevalent. You know, if all organisms alive have this kind of a molecule, but some organisms have this kind of molecule, that one should maybe be more ancient. There's no evolutionary branch, is what I'm saying. So this is the ribose. The anomeric carbon is carbon 1, 2, 3, 4, 5. That's why this way would be the 5' end, and this way would be the 3' end, and DNA and RNA are copied from 5 to 3. Right, or synthesized 5 to 3. When you read it, it reads the other way. So this is a ribose, and this right here would be what we call a 2' deoxy ribose. You know, carbon 2, there's no oxygen, right? So what would come first, synthetically? Do you remember that sugars are built biochemically, starting with, let's say, glyceraldehyde, right? And if you know something about photosynthesis and the Calvin cycle, then you could figure out how sugars are actually created and combined there. It's almost like the Kiliani-Fisher synthesis, where you're adding one carbon at a time. So you're adding a carbon and an oxygen, carbon and oxygen, carbon and oxygen. So if that's the way you're synthesizing this, on the way up here, adding a carbon and oxygen at a time, then what's first? What would come first? The ribose that has an oxygen per carbon, or a deoxyribose that is missing an oxygen over here? You would see, I think logic would tell you that ribose comes first and the 2' deoxy would be modified, rather than taking a whole new synthetic pathway to make the deoxy and add an OH. Like, that would be, why do that for just that one specific carbon, right? So then the question lies in why would DNA want to have a 2' deoxy? Is there any use for it, or is this just completely random? And it isn't completely random at all. At least organic chemistry and physics say it's not completely random when you look at it. Now, when I say not completely random, I hope nobody is thinking I'm pointing at saying something like God created it. It may be or may be not. I'm not commenting on this at all. What I'm commenting on is that when I let go of this pen, it falls to the ground because there is an attractive force between the pen and the earth. We could do the equations to show that. That's why I'm saying it's not random. I'm not saying that it's designed to be so. I don't know if it is or not or whatever. We're not getting into that, Reed. So when we're seeing there's a deoxy, is there a point to it? Not has it been created with a point, but maybe did time work it out that there's a point to it somehow? So what does the chain look like? We've just said that the repeating unit is a ribose with a nitrogenous base and a phosphate. And the chain for RNA or DNA is linked in a 5-to-3 kind of a way. That's how it's built, 5-to-3. Here is carbon 3, here's carbon 5. This, by the way, you're looking at is a nucleoside. You have to get this right. You have to get the language right over here. This is a nucleoside. Why is it a nucleoside? It's a nitrogenous base with a ribose. That's a nucleoside. That's not a building block of DNA or RNA. The building block is a nucleotide, meaning it needs to have a phosphate somewhere. So I'll put a phosphate right here. Now I've got a nucleotide. Do you see this? And if I now make a repeating chain over here, like think about amino acids and their repeating chain. It goes nitrogen, alpha carbon, carbonyl, linked to the nitrogen of the next amino acid, alpha carbon, carbonyl, linked to the nitrogen of the next amino acid. So what is the repeating chain here? It's phosphate, ribose, phosphate, ribose, phosphate, ribose. And like in your amino acids, there's an R group side chain, R group side chain. Here, the R group's the nitrogenous base, right? So it goes, if I'm going to show you a segment of RNA, it goes like this. It's some growing chain, and now I'm going to show the first phosphate. And now that oxygen is on carbon 5. Carbon 5 is connected to the ring. Sorry this is taking a few minutes to get to the point where I'm going to show you why DNA and RNA have some sort of a difference, right? But like, I need to make sure we structure the molecule right so that you can follow the story. So here is the nitrogenous base. And if this is RNA, it has its OH over here. And then an oxygen, and that oxygen is going to be connected to the next phosphate group in this phosphodiester linkage. What is this oxygen connected to? Once again, carbon 5. It just repeats. Phosphate, ribose, phosphate, ribose. So this oxygen is on carbon 5, and then carbon 4, part of the ring, with again a nitrogenous base. There's an OH over here. The rest of the chain keeps going. Do you see there would be an oxygen phosphate, carbon 5, right? Why is this called a phosphodiester linkage? You should have learned this in orgo. Any linkage like that should be called a phosphor anhydride. I'm pretty sure everyone in this room and online should have some clue of what an anhydride is, right? There goes that magical list again. Right, an anhydride? So that's called a phosphor anhydride. It's like an anhydride, but instead of carbonyls, it's phosphoryls, we could say, right? But I just mentioned a phosphodiester. I just wanted to make sure we don't think that the phosphor anhydride bonds in ATP are diester bonds. What would be a diester? Well, Here's a carbon, that's a carbonyl, and if they have an oxygen and then some carbon chain, that's an ester. What if on this side is an oxygen with another carbon chain? That's a diester. You see that carbonyl is a double ester? So wouldn't this be called a phosphodiester? Where this is some R group and this is some R group. Do you see that's a phosphodiester? It's an anhydride, it's a diester. Do you see this phosphorus is part of an ester this way and part of an ester this way? It's a 3 to 5 phosphodiester connection. So what we have over here is like a fragment, like a piece of an RNA chain. And over here I'm going to draw similar piece but of a DNA chain. So you have that oxygen and you have a phosphodiester linkage to carbon 5. Here is the ribose. This also has a nitrogenous base, except it doesn't have this oxygen right here. It does not have that OH right here. What it does is still have this oxygen. It has the phosphodiester linkage to carbon 5. From carbon 5, carbon 4, which is part of the ring. Over here, a nitrogenous base. How many of you have to keep looking up at my board to draw this? Just pointing something out. As I'm drawing this, you should already kind of be good enough with it that you don't have to keep looking up to copy and follow. If you are, then you're not in the right realm yet. That's okay. We're working on what you need to practice here, right? There's going to have to come a point sometime soon where you have no issue just drawing this on your own, not just being able to copy from a picture. So this doesn't have an OH on it, but this has an O connected to a phosphate and it keeps going, right? So I said a while ago that nature doesn't waste energy. This is a very important theme. It's like day one in Orgo 1. Nature dislikes charges, right? Nature doesn't waste energy. So if nature doesn't waste energy, why would it go through whatever process trouble it is to remove this oxygen to make it like this? Do you understand? Like, this is not the easiest thing in the world. If you had to do Orgo, what would you do to remove that oxygen? Maybe you would do PCC and then Wolff-Kishner. There's a way to remove that, right? But the point is you need to waste molecular energy. You need to waste oxidizing, reducing. You need to do whatever. Maybe you need to turn that into a leaving group and use a hydride to kick it off or something like that, right? So why waste the energy of doing that? Here's a little bit of a problem if you're a prokaryotic cell. If you're a prokaryote, then let's say you have a circular DNA, right? And where is that? That's in the cytoplasm, along with your RNA and everything else. And the cytoplasm is an aqueous environment, right? So this molecule, if you're a prokaryote, is living in the same environment as this, same environment as water. Why do we care? So what kind of bonds did we say this has again? It's a phosphodiester. It has ester. And if you have water with an ester at pH 7, you can cleave that ester, but when you cleave it, it won't turn into a carboxylic acid, because it's pH 7, it will turn into a carboxylate, right? My point is that water at a neutral cell condition will split that phosphodiester bond, cleave the phosphate, and break apart DNA. Scary proposition, right? Do you remember that when we talked about amino acids linking up in proteins, I said they are something called metastable. They're stable for now. They're kinetically stable because water's a garbage nuke. They're not protonated enough to be electrophiles. But if you leave a protein in water long enough, it will get hydrolyzed, right? Now, of course, proteins, do they really last your whole life? I mean, there's gamma crystalline in your eye that lasts your whole life. But most proteins kind of are made or destroyed like when it needs to be used. The DNA, on the other hand, it gets copied, but the original template, that needs to be perfect. You can't have accidental chops and mutations and whatnot, right? So this would be a problem because this is RNA, right? Here's DNA. So what do you notice? One way the eukaryotic cell avoids the issue of water splitting up DNA is by keeping DNA in a cell nucleus that doesn't have any water. So if you take DNA out of water, right, you're not going to have water chopping it so much. But you're going to go through any additional payment necessary. You know what I mean? Like, it's almost like if you have a house, you're going to put a burglar alarm, video cameras, whatever you're going to do to protect it. So one thing to do to protect it from self-splitting or splitting in water is to put it into a nucleus where there's no water. But here's the second thing you want to do to protect. Why are you going to remove that? Because you realize whether this is a good or a bad nucleophile, it's still 1, 2, 3, 4, 5 away from doing an intramolecular attack, right, and splitting that way. So you would go through whatever cost it is to remove that from the chain. And now if you keep DNA inside a cell nucleus where there is no aqueous environment, and you don't have a 2' oxy, 2' OH, this won't have any chance of splitting at all. There's also an interesting other experiment that if I have a beaker, remember again, water is a really crappy nucleophile. So if I put water with RNA into a beaker, it's really not going to split this easily. There's another reason why this is a phosphodiester bond and not just a regular diester bond. If this was just a carbon, then water would have an even bigger chance to attack over here. But because this is a phosphorus that has this other negative oxygen over here, you've got negative charge resonating through this entire area. The nucleophile's repelled even more by that negative charge. You see, it's even less electrophilic. Nonetheless, if you put water and RNA into a beaker, it will hydrolyze eventually. If you put water and DNA into a beaker, it will hydrolyze 100 times slower. And the explanation for that is the effective molarity, or the neighboring group effect, or the intramolecular attack. Intramolecular reactions are fast than intermolecular reactions, right? So these should kind of show you that RNA possibly came before DNA. How? How does this prove any of that? At least logically it proves it. You would modify this for a reason. You would remove that OH to protect, right? Now what about a bacteria? It doesn't have an issue having DNA inside its cell. E. coli, for example, has an entire lifespan of 18 minutes. If H2O has a terrible nucleophilicity and does, let's say, 1 attack an hour, or even 1 attack every 10 minutes, E. coli, what, has 1, 2 attacks. It's not the biggest deal. You're living 70, 80, 90, 100, hopefully 500 years, right? You're going to have a problem if every 10 minutes your DNA is splitting. When you put it back together, you might not put it back together right. So you're going to spend whatever energy it takes to make sure that it's not splitting that way. Do you see what I mean? Spend whatever energy to make a compartment called the nucleus. Spend whatever energy to keep water out of that compartment. Spend whatever energy to remove that OH from here. So this just makes very, very good logical sense. And I like the fact that we have to revert back to this list to make sure that it makes complete sense. What is the other difference between RNA and DNA? That RNA has A, T, C. I lied. It's Monday. A, U, C, G. And DNA has A, T, C, G. Is there a difference? Is there a reason why there's a U and a T? Here's a uracil. Make sure to copy this well. You've never seen it before. It has a portion of it that looks like a urea. Never seen it before. I gained a subscriber. Look at that. Someone likes bad jokes. For the past 3 years, I've been a dad, so I'm allowed to say dad jokes. Anyway, so this is a uracil, and here's a thymine. It's completely different. It doesn't look at all like a uracil. Okay, it looks exactly like uracil, it just has a methyl group. Now, thinking about everything you know about organic chemistry, what do you think would be synthesized first? Like if one of them is a modified version of the other, if like DNA is modified from RNA or RNA is modified from DNA, which one of these would you synthesize first? Like if you're synthesizing this from scratch, would you synthesize a U and add a methyl group, or would you synthesize a T and take off a methyl group? Probably, synthetically, you would synthesize and then add a methyl group, right? Like it wouldn't make sense to add a whole bunch of stuff. I guess unless you're talking about sculpture. You throw a bunch of clay and then cut it off, so not in all cases. It makes a lot of sense synthetically to make this first, and then you're going to go through a lot of energy to modify it, so why would you modify and put a T over here? Again, whatever DNA is doing is for protective purposes compared to RNA. See, RNA is just like a copy. DNA is the template, it's your blueprints, it's like your secret code, and RNA is just a printout, like a lab report of it. And if you print out and something happens to the printout, you're like, whatever, I'll print out another printout, right? But you want the original to be protected. You want like 12 copies of flash drives. Make sure to make 12 copies of flash drives, because if you only have one copy, your flash drive will fail. You'll be like most of the way through your thesis, and your flash drive will fail, and you have to redo 6 months of work. Anyway. So why put the methyl group there? Why go through the trouble? It has actually to do with neither of these as much as it has to do with the other pyrimidine. Which is that one, which is the letter C. C-U-N-T. Cut pie. Single rings, pyrimidines, right? Okay. So here is a little bit of a problem. Again, if DNA or RNA, let's say they are in an aqueous environment, pH 7.2 to 7.4, look what's interesting that could happen to C. What would happen to C if I put this into H plus and H2O? Amongst other things, one of the things that C can, because there's a lot of things, you can pronate this, you can pronate that, you can pronate a bunch of things. But one of the things you could do is these electrons could go here, and that double bond could grab that H over there, right? In a very enamine kind of an attack. And the mechanism begins by creating this. And do you realize that's an imine type of a bond? The positive on that gives that tons of carbocation quality. And water is great to attack a carbocation, so it attacks proton transfer, kick off the NH3, leave the O. What the hell does this turn into? Do you remember you could hydrolyze an imine and the nitrogen is gone and an oxygen replaces it? And when that happens, won't a cytosine become a uracil? Could you keep that in your mind for a second? We're not there yet. This is just like, oh shit, we have a problem. We have a cytosine uracil problem. Now imagine you had a secret code, you know, like the password to get into your own house or your own bank account. Better, forget your house, bank account. And the password was CUGACCGU, right? And you kept it in your wallet, which was an aqueous environment with acid. And then it's time to go and withdraw money from your bank account. You know, you've been saving it for your whole life. And you take out the note, but, you know, it's an aqueous environment acid. So one of the C's became a U, and you don't remember which one, because if you didn't memorize it, you wrote it down. That's why you kept it in your wallet. So your code is CUGAUCGU, right? Let's remember that the C needs to get protonated and water needs to attack, but at a pH of 7.2 to 7.4, there's not a ton of acid, so it doesn't happen for every single C. It might happen every now and again for a C. So like if you keep the note in your wallet for days, maybe one of them changes to a U, and you're staring at this and you're like, I know there weren't that many U's. And you have no idea which U is the correct U and which U is a fake U. Do you see what I mean? Like, how would you know which U is the real U? Not like which U is the real U, but like which uracil is the real uracil. And how about, what if you gave an ID card to the real uracil? What if before you put it in your wallet, you color-coded some of the uracils orange or something like that? Then when you read this, you'd be like, haha, I knew it. That one's not orange, right? So that's fake. That must have been a C. Well, instead of like color coding it, what if you gave uracils a little tag, like a methyl group? Called it a thymine. Okay, that's the only difference between a U and a T is the methyl group tag, an ID card. Then when you're reading your code again, you realize that anything that's not a T, any U, is really a C that transformed. So we have repair enzymes in our cells that read our DNA all day. And whenever they get to a U in the DNA, they say that's not supposed to be there because the real uracil is a thymine with an ID card. So they could cut out the uracils and put in cytosines instead. Do you see, without the ID card, how would you know which uracil to cut out? Which U is the real U, is what I'm saying. You would have no idea if this is the U or that's the U. Enzymes don't know anything. They just, you could give them a function to do and they'll do that function, perhaps. Isn't this like amazing? If you don't find this like incredible, then you're in the wrong field, right? No, really, it's like, imagine being in fashion and somebody's like, whoa, they used like snake skin and made these boots, and you're like, whatever. I'm not sure you should be in fashion, you know, like, you could either be like, I hate them or I love them, but like, I'm indifferent. It's like, I don't know. I've had students that ended up in amazing like medical or whatever schools, and you could see the almost religious experience when they see what's going on here. They're like, this is crazy. How seems very designed. Perhaps it isn't. Perhaps just time design. I don't know. Nature finds the way to avoid wasting energy. So eventually, kind of like our own brains make shorter synaptic connections and so on, it's the same exact thing. Eventually, things that didn't have this system to be able to repair ended up dying out because you have less of a chance to correct mutations and problems. But when this pops up, now you want to ask the next question, of course. If you could correct it this way by putting in a methyl tag, why not do that to RNA also, right? And the answer is nature doesn't waste energy. If RNA is only living long enough to go from your nucleus as a copy over to the ribosome, what is it, living a minute, 5 minutes, 10 minutes? Are you really going to waste the energy to replace every single uracil with a thymine, do chemical modifications for the chance that maybe one RNA? Imagine you have like some sort of like a factory, like, I don't know, you're the Ford factory, you're producing cars. If something is going to go wrong with every single car, you're going to change your machinery. But if something goes wrong with 1 out of 100 million cars, whatever, you'll take the loss on that one car, right? If it's DNA that lasts your whole life, you don't want anything going wrong with that. You're going to spend whatever money. But RNA is like typing out, is like printing out a lab report, right? Now imagine you print out a lab report. Are you going to take that lab report and put it between plastic and laminate it, put it on a wooden plaque and hand it to your professor to make sure nothing went wrong with that lab report? It's like, whatever. If something goes wrong with the lab report, I'm sorry, professor, I'll print out another one. I wasted the 10 cents on this piece of paper. Do you know what I'm saying? But like DNA, you'll laminate that. You'll keep it protected in a vault, which is the nucleus. I definitely want to try to get through. I'm going to do my best to have one more meeting. For those of you that are watching this online, our last meeting for the summer session is supposed to be this Wednesday. I'm going to do my best to make that happen because I have a lot to do, and I'm trying to put together the groups for the fall and finish up my summer course. So there might be a chance that I email on Wednesday morning and say I actually need the time today to finish everything up. I'm going to do my best to do this, to do this lesson on Wednesday, do our last lesson, because what I want to do for sure before we finish this summer program is the Sanger dideoxy method for DNA sequencing. We learned how to do a peptide sequencing, at least start to play that game. This is so important. Sanger won the Nobel Prize. That is one of those things that there's an ulterior motive over here. When you learn that, you realize how brilliant it was when it hits you, how brilliant it was what Sanger did to figure out how to sequence. But he figured out this method based on how DNA copies to stop it copying in certain places. And from there, you could figure out size of fragments and use gel electrophoresis to figure out what came in which order. This is more complicated of a theory than amino acid sequencing because there's such a variety to amino acids. But there's only A, T, C, and G. So you couldn't use the same method like chymotrypsin to chop at every A because you'll chop everywhere and you won't know anything. So this dideoxy method was brilliant, but it was brilliant for another reason. It made us realize that you could stop DNA copying in a particular way, and you could apply that to medicine. Because a virus infects a cell, takes over your cell machinery, so you can't stop a virus the way you could stop a bacteria. We could stop a bacteria with antibiotics because a bacteria has its own cell machinery. A bacteria has its own ribosome. And a prokaryotic ribosome is different than a eukaryotic ribosome. So we can find a drug that blocks a prokaryotic ribosome rather than a eukaryotic ribosome, and you stop the bacteria, you don't stop yourself. But when a virus infects your cell and uses your machinery, you're not stopping your ribosomes, you're killing yourself, right? But then they realized by using a method like Sanger dideoxy sequencing, which the overall idea is it stops DNA copying in certain ways. You could apply that to infected viral cells to stop DNA copying in those viral cells, so you get antiviral medicine from the Sanger dideoxy idea. So I want to teach us that well so that you can click into what antiviral medicine is, like AZT, acyclovir, and many others. So to do that, we have to first figure out how DNA is synthesized. So whether it's DNA or RNA, it is synthesized in a 5' to 3' direction. Let's start with the simplest connection between nucleotides, a dinucleotide. Make a simple dinucleotide. So if I want to connect A to, let's say, C, and make an AC dinucleotide, right? I'm going to have to start with a nucleotide. Let's do DNA. It doesn't matter. Whether it's DNA or RNA, it's the same exact idea. So here will be the nitrogenous base. I'm just going to put a circle here and write A. I don't want you to focus on I don't want to draw out what A is and then you focus on its structure rather than the general idea here. So whatever A is, you could put a nitrogen there and fill out the rest of A. Okay. And then, of course, if it's DNA, we'll have an OH over here. We'll have a carbon with an O, P, O, double bond O, O minus, minus. That's your A. And we're going to react that with a C. So this will have a nitrogenous base, that's a C. OH, carbon O, P, O, double bond O, O minus, minus. And if I'm saying that it's A plus C makes an AC somehow, and it's synthesized in a 5 to 3 prime direction, right, then this end is a 5 prime and this end is a 3 prime. That's like, for example, amino acids in the ribosome are linked together from the N terminal to the C terminal. And when I draw an amino acid chain, if I draw alanine, phenylalanine, tyrosine, isoleucine, right, then this is the nitrogen side and this goes to the COO minus side, right? Like that's, it's synthesized the N to the C direction. These are synthesized in the 5 to the 3 prime direction. So what we are saying is that it's going to feel a little backwards here. But if it's synthesized in a 5 to 3 prime direction, then I'm actually going to connect the 3 to the 5 of the new one. That sounds weird because I just said it's synthesized 5 to 3. Why am I doing 3 to 5? But look, I'm starting with this thing that has its 5 prime end and its 3 prime end. And if I make a link to this, now I've created a new 3 prime end, right? So you see I'm continuing it into the 3 prime direction, right? So what does this AC thing look like? I really hope you're paying attention. If you are, you're going to really like this. Assuming you know what I taught you in Orgo. We did enamine to imine, and then hydrolysis. Many of you follow that. So that's the level I'm talking about of knowing what we've done in Orgo. Basically the list, acid chloride and hydride thioester. So this is what this would look like. And this oxygen from the nucleotide, what do you call this nucleotide? Adenosine monophosphate, 2' adenosine monophosphate, right? So adenosine monophosphate is a nucleotide, right? What's adenosine diphosphate? It's a nucleotide plus phosphate. Or it's a nucleoside diphosphate. I have an adenosine connected to a POPOP, right? This would be a triphosphate, right? This would be ATP, adenosine triphosphate. What do I call it? A nucleotide triphosphate? Then I messed up the language. A nucleotide already has a phosphate in it, right? I'm trying to point out that ATP is not a nucleotide triphosphate. That would make it 4 phosphates. A nucleotide has a phosphate. That's a nucleotide with a phosphate. That's a nucleotide with a phosphate. If I want to call ATP something, I'm going to call it a nucleotide diphosphate, which is weird. I should just call it a nucleoside triphosphate. Please make sure you know the language difference between side and tide. Side has no phosphate, tide has 1. Okay, so that oxygen is connected to a phosphate through a phosphodiester. Now that other oxygen is part of carbon 5, right? Witnessing this. So we have that oxygen is this right here. And then we have carbon 5, then 4, then this whole ribose scenario, OH, and then this is the nitrogenous base, that is the letter C. Okay, for my Orgo people. What is the direction of the equilibrium? Because I put that arrow over there, but what's the direction of the equilibrium? Where am I making a bond? I'm making a bond from this kind of a group to that kind of a group, connecting them, and here's the bond. What is it? We could summarize this as an alcohol. And look at what this is. This is a phosphomonoester, and over here it's a phosphodiester, right? The real difference here is instead of a P double bond O, O minus, instead of a double bond O, O minus, so I have a double bond O minus, you are creating an ester. What's the direction of the equilibrium? We think it's from carboxylate to ester. So I'm pretty sure it's from ester to carboxylate, right? Do you see what I'm showing you? So this is a problem. You're about to see why DNA is synthesized from the 5' to the 3' direction. It is a problem because you can't do this. Actually, you know what's favorable? A plus C makes AC is not favorable. What's favorable is water chopping AC into A and C is favorable. Haven't we just said this before? That DNA is favorable to be chopped energetically. It'd rather go downhill. Instead of an ester bond, a carboxylate is lower in energy, right? We can make the arrow go the other way by introducing some leaving group. And that's actually what happens. The leaving group's not a water. Here's what the leaving group is. So our first molecule is going to be the nucleophile. And the next incoming molecule, the one that's going to become the new 3' end, is going to be the electrophile. But as an electrophile, this is not good enough. Instead of a C, I'm going to add in a CTP. This should not be very, very scary. Please understand what the triphosphate moiety that I'm drawing over here, look up the word moiety. What this triphosphate moiety is. It's a carbonyl kind of a looking thing. It's phosphoryl. It's for real. It's a carbonyl looking thing, oxygen, carbon. So that's an anhydride. And that's another anhydride. This is just 2 phosphor anhydrides, right? In fact, let me describe this. This phosphorus is part of a phosphor anhydride. This phosphorus is part of 2 phosphor anhydrides. This phosphorus is part of a phosphor anhydride and a phosphoester. Chopping this bond is more energy releasing than chopping this bond. Chopping this bond goes from an anhydride down the list to a phosphate. Chopping this bond is from a ester to a phosphate, right? Okay, now if you process this, if this is the nuke, you looking? If that's the nuke, and that nuke attacks over here, and you kick that off, actually question, should I kick the electrons up to make a TCAI and then kick that off? Or am I doing the correct arrow push right now? Let's take a vote. Who thinks I'm doing the right arrow push right now? No hands are up. The room is not confident in my answer. What does TCI stand for? Tetrahedral. What is the shape around this? Tetrahedral. It's already tetrahedral. You don't need a tetrahedral anything when it's already tetrahedral. See, this is what you need a TCAI for. Something that starts off trigonal-planar. This is a correct arrow push. Do you realize what we're doing right now? Do you realize I'm reacting an anhydride with an alcohol? It's basically like making aspirin. Do you remember aspirin with salicylic acid and acetic anhydride? Not only did you actually synthesize this in Chem 1, but last semester we did the whole mechanism and everything. So is this legal now? Will this go in this direction? Absolutely, because if you're going from an anhydride down to an ester, that is the correct direction, right? And look what else you kicked off. You kicked off a diphosphate. You see that diphosphate leaving group? We call this diphosphate pyrophosphate. Sounds like fire. And of course, water can split that pyrophosphate even further, right? Okay, here's a little bit of a problem, a conceptual problem. How does this alcohol know to attack this phosphorus and break that bond, How come it doesn't attack this phosphorus and break that bond or that bond, or attack this phosphorus and break other bonds? How does it know? The answer is it doesn't know anything. These molecules, actually, I'm wrong. I don't know. I don't know if it knows. I don't think it knows. But here's what is necessary. This is still a garbage nuke. This is still an ROH trying to attack a phosphorus that has negative charge resonating through it, so it's going to be repelling that phosphorus. So you need an enzyme to lower the activation energy so that this could attack over there. And that enzyme is called DNA polymerase or RNA polymerase. Unless you did it in a lab and you called it Taq polymerase, but that's just a type of DNA polymerase. And what does the polymerase do? It grabs this and grabs that, and it helps make this electrophilic. How does it do it? It often uses various ions that we might have available. For example, a magnesium 2+ ion. Can you imagine if the DNA polymerase enzyme up here, over here, places a magnesium 2+ ion? Wouldn't that attract the electrons over to that magnesium? And therefore, this would have negative resonance all over there, but this would actually be a little positive. Wouldn't that attract the alcohol electrons over there rather than over there? So the enzyme is not doing something that wouldn't happen on its own. This would attack there or there or there. It would just take, I don't know, 1 million years for the crappy nuke that's an alcohol to find its way to attack that. The enzyme's just lowering the activation energy barrier, right? The enzyme's not changing. Reactants want to become products. Do you see? By the way, have we learned polymerization before? Have we learned about step growth and chain growth polymerization? Do you remember those polymerizations where they're the same kind of molecules, but like, it's like a domino effect. I got hit by a domino, so now I am the hitting domino kind of a thing. Those are the chain growth polymerizations. Isn't this so chain growth? The 3' end acts as a nuke, kicking off a pyrophosphate, giving you a new 3' end. And now that 3' end is going to react with another ATP or CTP or GTP or TTP. So look, the electrophile, this molecule's the electrophile, becomes the new 3' end, which is the nucleophile. And it's going to attack another ATP, CTP, GTP, or TTP, which that thing will become the new 3' end, which will be the new nucleophile. Do you see the ATP, CTP, GTP, or TTP is the electrophile, but becomes the new 3'? Do you see why it's going from 5' to 3'? Do you see why the synthesis is in this direction? If I want to synthesize a DNA chain, there's a couple of ways to do it. There's an organic chemistry way to do it, and then there is a kind of more natural way to do it. You're still using a little bit of like orgo techniques, but you're hijacking the enzymes. So if I take a piece of DNA, somehow I get a piece of DNA and I put it in a test tube, and I put in DNA polymerase and all the ingredients that would happen in a cell, that's more of the natural way to do it, even though it's happening in a test tube. And then there's ways that I could modify the nucleotide to put specific leaving groups and nucleophiles on it and do a totally different synthesis of this. I'm talking about the more natural way. For example, in the end of the chapter we just finished, there was amino acid synthesis, like protein synthesis from amino acids. So you could synthesize proteins by using the natural way, the ribosome, tRNAs, that's the natural way. Or you could use these protecting groups that I mentioned last semester, like the Bach and the Z protecting group, and use DCC and DCU. Those are specific reagents. You weren't here last semester, read in the textbook. Those are two different ideas. We're talking about the slightly more natural way. It's still in the test tube. So you need 7 lucky ingredients, basically. And I can't synthesize any DNA that I want. I'm going to have to actually only copy DNA. So we're talking now about how to copy DNA. You need the DNA piece. You need the actual piece of DNA you're trying to copy. Besides the DNA, you need something called a primer. That's like a little snippet of DNA. and it's a little piece of DNA that's complementary to this DNA that you have. I'll talk about that in a second. And then besides the primer, you need deoxy. We're doing a DNA copying here. You need the DNTPs. The deoxy, listen to the language, deoxy nucleoside triphosphates. Why? Because the triphosphate is the thing that gets attacked and becomes the new end that does the attack, right? So what are the DNTPs? DATP, DCTP, DGTP, DTTP. How many ingredients so far? 1, 2, 3, 4, 5, 6. I'm missing something. What do I put in there? The enzyme. We just said it. DNA polymerase. The polymerase enzyme. I'm about to describe something very simple. It's way more complicated than what I'm about to say, but it kind of works in this way. The polymerase enzyme, again, is not smart, I don't think. It might be. If you have a conversation with it, let me know how that goes. But here's what typically happens. You've got DNA that unwinds in a place called a replication fork. So your DNA unwinds, and it copies the strands. One is called a leading strand, one is called a lagging strand. One thing we need to know is when you have double-stranded DNA, one side goes from 5 to the 3' direction, and the other side goes from the 3 to the 5' direction. They're complementary, and they go in opposite directions. So DNA is synthesized from the 5 to 3' direction. We just saw that. So that means the polymerase enzyme would actually be reading a piece of single DNA strand backwards. It would be reading it from the 3 to the 5 and building the complementary strand from the 5 towards the 3. Right? So if you have a piece of DNA, one is from 5 to 3, one is from 3 to 5, right? And there was a problem with one of these strands in trying to copy it. So if I'm copying this strand, the copy is going in this direction from 5 to the 3' direction, right? Following? On this one, we have a little bit of a problem, because imagine this animation, right? Imagine you've got a piece of DNA, and then it opens up at the end, and then it keeps unwinding, right? So then the unwinding gets bigger and bigger and bigger, right? When this opens up, let's say this chain is 3' to 5'. You're going to start copying in this direction, and as it gets wider, you'll just keep copying in that direction, right? But on the other way, you have to copy in this direction. So once you copy in this direction, when the opening gets wider, you have to like disattach and reattach and keep copying this way. So there's these little fragments called Okazaki fragments, named after a Japanese scientist who found them, figured them out, blah, blah, blah. So there's that problem. We're not going to really have that problem in this tube because we're just going to have one strand and not two strands separated. But I'm trying to give you a little bit of a visual here. How does the DNA polymerase work? On some level, it is able to recognize the region where double-stranded is converted into single-stranded. When it finds the location where double-stranded becomes single-stranded, it knows that it's at the replication fork. It knows that it needs to sort of attach over there, and it knows that it needs to complement one of these single strands. Do you follow the idea? So how do we replicate that in a test tube? We don't have a replication fork, we just have a piece of DNA. What if this piece of DNA from the 3' to 5' direction was the letters A, T, T, G, C, T, A, C, C, right? Imagine we threw in there a little primer, a little piece of DNA that started us off, that started off the copying. Imagine from the 5' to the 3' direction, this little primer had the letters T, A, right? Then wouldn't that little primer stick right up here? 5' to 3', T, A. And now you could confuse the DNA polymerase enzyme because this is a place where double suddenly abruptly becomes single. and it thinks it's at the replication fork. So what it needs to do is continue this chain in the 3' direction. So it's going to fit in whatever's complementary to the G, which is a C, whatever's complementary to the C, which is a G, and keep that going. So it's very necessary to add in a primer. Now we get into this funny area of like, where the hell do we get the primer from? Because if you get the DNA, okay, I need the beginning as a copied piece. Like, how do I get that? If I could only copy, how do I get a copy of, like, where's the first copy come from? Well, it turns out we have synthetic techniques to be able to create a primer. So in a laboratory, we'd call up a company and say, I need a primer with these letters in this order. And the company would synthesize it. It's pretty cheap. It's like $1 a base pair or something like that right now. And they'd send us a sample with dry ice and whatever, and make sure it's cool, refrigerated. And we'll have our primer. But of course, how did they build the primer? Well, they didn't do it in this 7-ingredient biological type of a way. They did it with this chemistry called phosphoramidite. Which is just like modifying nucleotides to put leaving groups and electrophilic positions and nucleophilic positions so that you could just throw them in the flask and then they just kick each other off and create a chain. Like, you realize you could. It's almost like what I'm trying to say is, how do amino acids combine and make a peptide bond? Well, this is going to attach to a tRNA, that's going to attach to a tRNA and a ribosome, they do all that. Well, you can just do orgo. If I want this to connect to that, I will turn that into an acid chloride, throw them together, they'll just attack each other, game over, right? So the phosphoramidite is like putting on artificial leaving groups, and it just, you can look that up, it's not that crazy, although it does involve a solid state synthesis, which you should read the chapter we just got out of Merrifield solid state synthesis too. I'll describe that in biochem when we get to it. So by the end of this lesson, which we're already over time, I just went over time in both lessons. That's nice. You should have a sense of what it means to copy from the 5' to the 3' direction, what the nuke is, why it works energetically, what these ingredients are all about, and how I could copy a strand of DNA. And if you know how to do that, then we're going to say, what happens if instead of deoxy ATP, like what does deoxy ATP look like? And of course, I'm talking about 2' deoxy ATP. Instead of deoxy ATP, there's the letter A with O, P, O, P, O, P, O. Instead of 2' deoxy ATP, what if in this flask I put in 2,3 dideoxy ATP? What if I put in a molecule that had no OHs on the bottom? When it came time, so what would go next? The A's 3' OH, right, would be the nuke to attack a CTP, right? So it would attack over here, kick off that, and that would be a C in the chain. And then that C would attack a GTP, and then that GTP would attack an ATP, but the ATP would be dideoxy, so the ATP would have no nuke over here. So the chain would stop growing at the A. Does that make a little bit of sense? What if I put in ATP and dideoxy ATP, a mixture of the two? Now sometimes you're going to get a regular A and the chain will keep going, and sometimes you're going to get a termination. If you put that one in, the chain will stop growing at that point. It is that idea that led Sanger to develop this thing called the dideoxy method, where they put some dideoxys of each one, did 4 different experiments, and were able to make it terminate at all positions. Then did electrophoresis to separate the chain sizes to know what came first, what came next. We'll deal with that in the beginning of the next lesson, which I hope is this Wednesday. Thank you for participating.